Welcome to our State of the Science snapshots from the Science and Technology team at the Good Food Institute. I'm Liz Specht, Vice President of Science and Technology, and I'll be joined by several of my colleagues to highlight the most exciting developments happening in the science and technology ecosystem for alt proteins. These snapshots are presented 3 times a year, and each snapshot covers noteworthy developments over the prior 4-month period. This field is moving far too quickly to cover all of the advances in a comprehensive way in a short video like this. So these snapshots really serve as a highlight reel curated by our subject matter experts and team leaders. We've got a busy rundown from an exciting start to the year, so I'm going to jump straight to our agenda for today. First, I'll hand over to my colleagues, Dr. Samantha Riches and Amy Huang, to talk about advances in the alternative protein scientific funding and training ecosystem. Then we'll turn to our technical subject matter experts, Drs. Priera Panescu, Adam Lehman, Faraz Harsini, and Claire Baumkamp, to talk about what has caught their eye over the last few months in terms of new research successes and noteworthy publications in each of their zones of expertise. I'd also like to give a shout out to my colleague Renee Bell, who's advancing the slides for us and helped to organize and manage all of this information to make these snapshots possible. With that, I'll hand over to Samantha.
Thank you so much, Liz. Following our highlights in the previous States of the Science snapshot, we're continuing to see positive signals from the US government regarding its support for alternative protein research. In March this year, following the publication of the Executive Order on the National Biotechnology and Biomanufacturing Initiative, the White House published a visionary Bold Goals report. The report looked at how biotechnology and biomanufacturing R&D can address societal challenges, with a number of sections being led by different government agencies. Through this, the report identified clear themes and a series of ambitious goals to address climate change, food and agriculture, supply chain resilience, and health, as well as looking at cross-cutting advances that are needed. We were delighted to see that alternative proteins were explicitly highlighted within the goals addressing both food and agriculture as well as climate change. These sections of the report were led by the US Department of Agriculture and the Department of Energy, respectively. The contributions were made from other agencies with interest in this space. The R&D needs highlighted covered a range of important issues, from structural design and food architecture through to research to inform regulatory efforts and safe commercialization of new products. We're now eagerly anticipating the publication of the Strategy and Implementation Plan, which is being led by the White House's Office of Science and Technology and Policy and is due later this month. Over the last 4 months of this year, there have been a number of exciting new research investments and commitments in alternative proteins. I'd like to take this opportunity to highlight just a couple. GFI grantee Marianne Alice from the University of Bath, UK, was successfully awarded a new £12 million investment in cellular agriculture manufacturing. This investment will focus on usable and scalable manufacturing systems and brings together a number of alternative protein research hubs in the UK, namely Aberystwyth, UCL, and the Royal Agricultural University. The centre itself was funded through EPSRC's call for manufacturing hubs for a sustainable future. In addition to this, the Israeli Institute of Technology, Technion, has committed to a $20 million sustainable protein research centre. While this centre is still in its initiation stages, Technion has outlined a series of ambitious plans to coordinate efforts across departments for a multidisciplinary, multi-platform centre. Once established, the centre will seek to build on existing research efforts by recruiting new faculty members in the field, purchase and maintain new equipment, and fund new and additional research. GFI was able to play an active role in the development of this centre by working closely with the managing team, providing knowledge and guidance about white spaces and identifying Technion's strengths. We are really excited to see the new research and partnerships that come from this. Finally, a number of new alternative protein-focused calls and initiatives were launched during this time. For example, the Science Foundation of Ireland launched its Future Food Systems Challenge, which explicitly called out alternative proteins within its highlighted topics. Applications closed in May and decisions are due this month. Likewise, USDA's AFRI Foundation and Applied Science Programme was launched in February for awards between $50,000 and $800,000. Alternative proteins were explicitly called out within the Novel Foods and Innovative Manufacturing Technologies Programme area. This call remains open with a close date of September this year. Finally, I'd like to highlight the Israeli government alternative proteins workforce training tender. This open call for training with an emphasis on the food tech industry follows a study by GFI in collaboration with the Ministry of Science, Innovation and Technology and the Ministry of Economy, which identified training needs within the alternative protein workforce. This call closed in May and decisions are due in July. And with that, I'd like to hand over to my colleague Amy, who will give an update on alternative protein talent ecosystem.
Awesome. Thank you so much, Sam. Leading climate think tanks are beginning to illuminate the economic growth and job creation potential of the alternative protein sector, which, as it turns out, is quite substantial. In April, ClimateWorks Foundation and the Global Methane Hub released a global innovation needs assessment in which they analyzed the impacts of various interventions for reducing the methane production in our global food system. In the report, they projected that the alternative protein sector has the potential to generate 83 million jobs by 2050, which they estimate to be 2/3 of all jobs created by methane innovation. Alongside this job creation, they estimate that economic value generation is also dominated by alt proteins, representing roughly 98% of total value generation by 2050. These projections give us reason to be optimistic that this sector is well poised to drive economic growth and job creation into the future. But of course, whether these projections actually materialize will largely be dependent on the amount of public and private investment dollars that pour into R&D, physical infrastructure, and workforce development programs. Alongside these forward-looking projections, we do know that there are major technical talent constraints in the field today. In March, we compiled results from a recent survey of alternative protein startups and identified fairly universal challenges with securing the right scientific and engineering talent to power their teams. When we asked startups to select the top technical skills and backgrounds missing from their teams today, we found that these general or cross-platform skills and backgrounds appear to be the most needed. These skills include disciplines like food science and meat science, process development and scale-up, manufacturing, product development, and food safety, which all have a really wide range of applications across the spectrum of alternative protein technologies. When we ask these same companies to predict the top technical skills they anticipate needing most 5 years from now, skills related to manufacturing, process development, and scale-up, product development, and automation floated to the top. Of course, beyond these cross-platform skills, specialized platform-specific skills and backgrounds are also needed, but I won't dive into that here. If you're interested in learning more, I do encourage you to dive straight into the full analysis on our blog. In the meantime, I'll flag one major key takeaway, which is that these awareness and training gaps will not close themselves. Startups indicate a super healthy appetite for partnering with academic institutions to build the relevant programs and pathways. So we enthusiastically encourage any colleges and universities interested in co-developing these programs to train the future food workforce to reach out. We are also seeing a growing number of institutions around the globe rising to this precise challenge by deepening their offerings related to alternative protein education and training. To name just a few notable developments from the past few months, Wageningen University recently launched a short course on the protein transition, presenting diverse perspectives on alternative protein technologies. EIT Food now offers a focus area track on protein diversification as part of their Sustainable Food Systems Master's Program. The San Diego State Alt Protein Project hosted the Alt Protein Summit, featuring panelists from across the academic, private, and civil sectors. And the Duke and Chapel Hill Alt Protein projects partnered to host Making Alt Proteins the New Default, a student-led conference in the Research Triangle Park with over 200 registrants that packed the day with panels, student pitch competitions, keynote speeches, and networking. And all of these activities, formal and informal, serve to build a better equipped talent pool to drive forward alternative protein innovation, such that the field has the person power it needs to meaningfully address society's most pressing challenges. That is all for me for this period. I'll turn it over to my colleague Pri for an exciting crop of updates on the frontier of plant-based meat science.
Wonderful. Thank you so much, Amy. I'll highlight some of the technical developments for plant-based proteins and other ingredients from the first 4 months of 2023. For a plant protein source to become a popular choice amongst growers, it must have a proven track record of good, consistent yields, resistance to droughts and pests, and high demand. Understanding more about the traits of crops that are used for plant-based foods can help ingredient producers choose their raw material sources and crop breeders to more strategically create resilient crops with strong potential for commercialization. GFI grantee Dil Thavaraja and colleagues evaluated the traits of 25 commercial pea cultivars and compared some of their protein content, amino acid content composition, and digestibility. The left graph here demonstrates some of these results, which generally show good traits overall, with some variations between cultivars that could be interesting to breeders. Additionally, researchers from the University of Alberta and Agriculture Agri-Food Canada evaluated 7 pea genotypes and demonstrated a range in functional performance and digestibility that could be attributed to the differing protein fraction ratios in the pea cultivars. In the middle graphic of this chart, we can see the emulsification capacities of the 7 pea cultivars varied significantly. Finally, in a huge collaboration, researchers from Aarhus University, the University of Helsinki, and the University of Reading, amongst other universities, mapped the enormous faba bean genome, which is 4 times bigger than a human genome. As an example of the information that can be drawn from the faba bean genome that they mapped, researchers dove into understanding what genes cause the difference in cultivar seed colors. The figure on the right here demonstrates the 2 genotypes and their visual differences in the seed center color, as well as a phylogenetic tree demonstrating the relationships between a pea gene that causes a similar type of seed pigmentation and the activities of similar genes found in the faba bean genotypes, allowing them to identify which faba bean genes relate to seed color. The genome data can also be used to evaluate the connections between other crop genes and other important traits like faba bean crop yield, resistance to drought and pests, and improved nutritional traits, expediting crop breeding efforts. There are a number of different ways to process a crop into its components. And in the beginning of 2023, researchers explored how these fractionation techniques affect the ingredient output's environmental footprint, cost of production, and estimated revenue. First, researchers from Wageningen University reviewed mild fractionation techniques, which is a gentler version of extensive wet fractionation, including a dive into various fractionation methods and their environmental impacts. On the graph on the left here, we can see that when lupin and pea were evaluated, any fractionation of the original crop resulted in more greenhouse gas emissions. Simply producing a flour by milling the crops only increases emissions a little bit, but once other fractionation methods are applied, we see that dry fractionation followed by mild fractionation and finally wet fractionation techniques increasingly impact greenhouse gas emissions. Additionally, researchers from Intelligent Inc. released a very interesting techno-economic assessment of soybean refineries, where 2 refineries were modeled. Both processed 80 metric tons of raw soybean per hour into soybean oil, lecithin, hulls, and 1 of 2 protein ingredients. In the first modeled refinery, the only protein ingredient produced was soybean meal, which is minimally enriched and typically used for animal feed. In the second refinery, they produced higher value protein concentrates and isolates that can be applied toward human food consumption. The techno-economic analysis demonstrates that while the upfront and operational costs for the second refinery are higher, the additional annual revenues resulted in a higher return on investment and reduced payback time, as shown on the graph in the right here. The authors state it is obvious from the results of the analysis that the co-production of soy proteins improves the economics of soybean refinery. They encourage people to test drive this model, which is available on their website, and the full report is available open access online. High internal phase emulsions have emerged as versatile tools for food formulators to form nutrient-dense materials. High internal phase emulsions are mixtures of oil and water phases that form tightly packed droplets. These droplets can be so tightly packed that they form semi-solid structures. On the left, we can see a recent exploration of a high internal phase emulsion of algae oil stabilized with water-soluble conjugates of pea protein isolate and inulin. The solution was rapidly mixed, and the pea protein inulin conjugates are able to stabilize the oil into a solid structure. We can see the resulting product doesn't fall when the container is flipped upside down, and this microscopy image also demonstrates the dense packing of oil droplets stabilized by the pea protein conjugates. The authors determined that the inulin conjugate to the pea protein helped the emulsions be more viscous and stable and elastic, making it a really good candidate for 3D printing. And they actually do demonstrate a few model food products through 3D printing this mixture. High internal phase emulsions formed by plant proteins can be used for a myriad of applications in addition to 3D printing. And researchers from the University of Connecticut published a great review on these applications, with the summary shown on the right here, and includes applications like fat substitutes, encapsulation and release of other ingredients like vitamins and minerals, and building porous structures. I'll wrap up plant-based developments with a few research projects focused on plant-based meat texturization with techniques other than extrusion. While extrusion offers a scalable texturization solution for the plant-based industry, novel methods definitely should be explored that can improve end product texturization, lower energy consumption, and improve costs of the process. First, researchers from Wageningen University demonstrated that when applying shear cell technology to pea wheat protein blend, shearing during cooling resulted in weaker products. The authors tested a range of processing conditions that varied rotational speed, shearing temperature, and shearing time. Some of the microscopy images of the products are demonstrated here, and we can see some subtle differences between the structures. Additionally, researchers from the University of Massachusetts Amherst and Zhejiang Gongchang University took a low energy approach to plant protein texturization as depicted on the right here. Low energy texturization methods that don't require large equipment could help lower the costs and improve the sustainability of plant-based meat production. In this novel approach, the researchers formed coacervates, which is just a fancy way of saying a soft material that is formed through interactions of oppositely charged molecules. In this case, potato protein and gellan gum. They then sheared the soft material to align it in a way that mimics muscle fibers before strengthening it with the gelation step. This simple method yielded a fibrous product that behaved similarly to conventional chicken during heating. And that wraps up plant-based proteins. With that, Adam will walk you through some exciting fermentation-focused updates.
Thank you so much, Pri, for those great updates on plant-based proteins. I'm happy to share with you some of the many advancements in fermentation-derived alt proteins during the first months of 2023. Much of the research on fermentation-derived proteins has been on the technical and biochemical aspects of their production. New research on mycoprotein earlier this year has focused on the nutritional and health outcomes of diets where mycoprotein is a primary protein source. In 2 research papers, both funded by Marlow Foods, makers of corn, researchers have shown that fungal biomass as a main source of protein is no impediment to athletic performance, such as the weightlifting evaluation shown here in figure labeled 1. Further, with a microprotein-based diet, there are improvements in gut microbiome health as compared to standard animal-based protein diets, particularly in the increases of beneficial gut bacteria population, shown here in number 2. Last on this slide, a published meta-analysis of nutritional studies on plant-based and mycoprotein containing diets showed that consumption of these alternative proteins rather than an omnivorous diet had statistically significant improvements in health markers such as total cholesterol, LDL, and triglyceride levels. New research is revealing the pathway towards better sensory experience with fermentation-derived protein products. Researchers at Lund University and Macarena published a manuscript exploring methods for protein extraction from fungal biomass and demonstrated the changes in protein extraction efficiency, emulsion, and foaming that can be brought about using different processing strategies. These strategies reveal the different ways that the same protein substrate could be used for many different food functionality applications. To date, egg white protein has been an important binder for the food industry and is even used in some mycelial protein products. Researchers are actively looking for alternatives to egg white proteins, but need to understand the properties that make it an effective binding agent in foods. Researchers at Cornell and the University of Leeds published a detailed study on the interaction between mycoprotein and egg white protein, and the authors identified how the egg white protein coats mycoprotein filaments and some of the changes in the interaction when the salt concentration and pH of the conditions are varied, both of which are relevant to food preparation and manufacturing. This will hopefully lead to new functionality for plant proteins to replace egg white protein. Next, genetics and omics approaches allow for insights into the metabolism of fermentation organisms and feedstock systems and can guide strain development. These developments will lead to better fermentation organisms that will improve the alt protein fermentation-derived space. In Figure 1, we're showing a paper that demonstrates the expanded metabolic repertoire of organisms. So scientists at the University of Tartu in Estonia and Lancetech have published a preprint that explored the proteome of gas fermentation bacteria in cells where C1 carbon metabolic proteins are knocked out. Interestingly, one of the knockouts affected the level of ribosomal proteins, which is vital for peptide synthesis and the growth of biomass. This study and others like it are important for shedding light on how these microbes are able to fix carbon dioxide and carbon monoxide and convert these gases into biomass. In Figure 2, we're showing Aspergillus oryzae as a well-established food fungi. Its ability to enhance the taste and nutritional value of its substrates make it an important fermentation organism. New work investigating A. oryzae fermentation of pea protein showed that the fungi upregulated the biosynthesis of sulfur-containing amino acids and remodeled the protein content of the fermentation system, the pea protein as well as the fungi. The analysis demonstrated how fungi could improve the protein profile of future alt protein products by providing amino acids that are relatively rare in plant-based protein alone. Comicatella pfaffii is a workhorse species for precision fermentation. Earlier this year, a series of genetic tools were made available along with a preprint manuscript describing their use. OpenPicia is an effort by researchers at VIB in Belgium to create a highly transformable chassis strain, which can also be used and distributed under resale or commercial license. These strains are complemented by a suite of open-source genetic elements that can be used to construct cassettes to drive the production of food ingredient proteins. To wrap up our fermentation section today, life cycle assessment can guide researchers to opportunities or challenges that maximize the sustainability of products in their manufacture. An LCA study from the University of Helsinki designed to understand the environmental impacts of animal-derived proteins, as well as a host of alternative protein sources, was published this year. The authors used a variety of environmental impact categories to gauge the sustainability benefits of fermentation-derived alternative proteins, like microalgae, mycoproteins, and microbial single-cell protein. The good news? Modeling each of these alternatives demonstrates their ranking above animal-derived proteins in environmental impact. By isolating each category, like eutrophication, global warming potential, and land use, the authors allowed for identification of opportunities to make fermentation-derived alternative proteins an even better choice for sustainability. For example, improving water use efficiency in mycoprotein production or reducing eutrophication of albumin fermentation are both ways to improve the sustainability profile of those products. Next, an international team of industry and university researchers published a study on the potential for a global waste-to-food fermentation industry. Within this study, they surveyed the potential large-scale feedstock options and microbial species ready to convert them to food-grade biomass protein. The authors identified the potential for conversion of some 2,500 megatons of glucose, or almost 4,000 megatons of glucose plus xylose available for fermentation. If we're able to expand the repertoire of protein-producing microbes capable of pentose sugar metabolism, the opportunities to valorize xylose sugar present an enormous opportunity for the alt protein space. And with that in mind, identifying new biomass fermentation species will be beneficial to diversifying our food system options and also increase sustainability. Research on Pleurotus austriatus, also known as the oyster mushroom, mycelium, the mycelium and not the fruiting body, shows that its promise for the fungi as an alternative protein source by demonstrating its ability to grow in liquid fermentation on hydrated wood fiber, a side stream of the paper and pulp industry. Thank you. And now I'll hand it over to Faraz for technical developments in cultivated meat.
Thanks, Adam. So I'm going to talk about 4 recent developments in cultivated meat field. So first, Japanese researchers have found a sustainable way to grow animal cells. They've harnessed cyanobacteria, which is a unique type of bacteria, to consume a waste compound, which is L-lactate, that can harm animal cells. They introduced an E. coli gene into cyanobacteria, enabling it to digest L-lactate. This method, by using bacteria to recycle harmful waste like L-lactate and ammonium, could pave the way for a sustainable and circular cell culture system. Next, a study has revealed the possibility of using nutrients from microalgae and cell-secreted growth factors as a substitute for a conventional grain-derived nutrients and FBS in cultivated meat production. This research showed that a unique blend of microalgae nutrients and cell-secreted factors could actually promote the proliferation of bovine myoblasts, which are basically a crucial cell source for cultured beef. This has the potential to reduce the environmental footprint and production costs linked to traditional meat production. The microalgae's CO2 absorbing capacities could also contribute towards carbon neutrality. This is the first study to report that myoblasts can proliferate sustainably using microalgae-derived nutrients and cell-secreted factors, providing a low-cost alternative for the production of cultivated meat. In this study, researchers created edible microbeads made from alginate, but coated with pumpkin seed protein that show promise for cultivated meat production. Alginates are basically natural and edible polysaccharide polymers derived from the cell walls of brown algae. The protein coating can stimulate the growth and attachment of cells. These microbeads, tested against 11 different plant proteins, showed superior ability to stimulate cell growth. With qualities similar to animal gelatin microbeads, these pumpkin seed protein-coated versions advanced the development of edible and animal-free materials for creating cultivated meat. And lastly, researchers have created a new way to produce 3D cultured fat tissue for food purposes. This technique overcomes issues like nutrient and oxygen delivery and toxic waste buildup. It involves growing adipocytes or fat cells in 2D, then combining them into 3D structures using binders like alginate. The resulting 3D fat tissues closely resemble animal fat tissue in texture. In addition, their fatty acid composition can be adjusted through lipid supplementation during cell culture. This affordable method can be applied to various cell types and species and can be scaled up for large production, making it a promising development for the future of cultivated meat. And with that, I'll pass it to Claire to talk about recent developments in alternative seafood.
Thanks so much, Faraz. I've just got a couple of quick updates on the latest progress in the alternative seafood space. So first up, one challenge for producers of whole-cut alternative fish, whether that be plant-based, fermentation-derived, or cultivated, is the unique geometry of fish muscle tissue, which is made up of alternating layers of muscle called myomeres and fat and connective tissue called myosepta. When looking at a raw salmon fillet, if you can imagine the alternating orange and white stripes, those are actually the myomeres and the myosepta, respectively. So researchers from the National University of Singapore recently published a paper on the use of 3D printing to create whole-cut plant-based salmon consisting of red lentil protein-based myomeres alternating with pea protein-based myosepta. Next, a couple of quick updates on cell lines for cultivated seafood. In March, the GFI APAC team hosted Dr. Shigeki Sugi for a webinar where he discussed his team's recent progress developing fat cells from Pangasius, barramundi, eel, and perch. The lines developed by Dr. Sugi's team have been banked directly through A*STAR's Crisp Meats program, and researchers interested in working with these lines will need to set up a research collaboration with Crisp Meats. And so if that's you, please just reach out to Dr. Sugi directly. In related news, researchers from the Czech Republic published a paper describing the development and characterization of 10 new embryonic lines from 2 species of killifish. Although killifish are not a high priority species from a food perspective, the availability of multiple embryonic lines from these 2 species may be helpful in improving our understanding of cell type specification in fish in ways that could be helpful for cultivated seafood. In January, GFI held a workshop where we convened a group of academic researchers and industry scientists to discuss the unique challenges associated with culturing fish cells and how the associated barriers to progress in cultivated seafood might be overcome. We had 3 main goals in convening this workshop. First, we wanted to make sure we understood the challenges facing the industry in as much detail as possible to inform the next steps GFI will take when it comes to cultivated seafood. Second, we wanted to increase connectivity within the cultivated seafood research community and provide another opportunity for collaborators to meet. Third, because we recognize that progress in cultivated seafood is going to require actions by many different players, we wanted this to be a forum where experts could bounce ideas off one another and come away with the best possible ideas for their own independent next steps. In total, 91 scientists attended the event, and we learned a lot from the workshop participants about the barriers they're facing and what they would like to see happen to address those barriers. I want to say we're so grateful to everyone who attended the event for showing up with a collaborative mindset and sharing their insights. So huge thank you to you. From those insights, we distilled out some major themes, which included the lack of validated antibodies and other research tools, the interdependence of multiple challenges in this space, such as cell lines and culture media, the challenges posed by our limited fundamental understanding of fish biology, and the need for more collaboration and research funding. What we learned from this workshop will inform GFI's seafood strategy for the remainder of this year and beyond, so please stay tuned for more updates. One thing we have coming up soon as a direct result of the discussions in and after the workshop is that we'll be piloting a new event series we're calling Fish and Chips, Collaborative Huddle for Ideation and Problem Solving. This will be a chance for researchers to get together in small groups and discuss the challenges they're facing in their research. So if you're a scientist working in cultivated seafood and would like to be part of the ongoing conversation, please consider joining us for that event on July 11th. Finally, in February, we published a new page in our series of technical deep dives, this one focused on cultivated meat end products. This new page goes into detail on a variety of considerations related to end products, including some of the unique challenges when it comes to cultivating seafood. In April, we published an update to our existing deep dive on cultivated meat scaffolding, which now includes a section specifically dedicated to scaffolding for seafood, as well as some additional seafood content woven into the other existing sections. These pages were a blast to write, and we hope you'll find them helpful. With that, I'll pass it over to Liz to close us out.
Thank you so much, Claire. That wraps our State of the Science snapshot covering January through April of 2023. Again, these highlights represent just a curated collection of the most noteworthy developments in the field from our subject matter experts, not a comprehensive list of everything that's happening. To stay up to date at a more detailed level throughout the whole year, get updates about all of our resources and publications by following us on social media and enrolling in our newsletters. And if you'd like a deeper dive on the science at any time, check out our detailed explainers, which link to tons of additional research papers and resources in our alt protein literature library. These snapshots and all of the insights, research, and resources that GFI offers are made possible by our donors. If you're interested in supporting more events like this and all of GFI's work to advance the alternative protein ecosystem, please contact philanthropy@gfi.org or visit gfi.org/donate. Thanks so much for tuning in.